Hey y'all, it's Miss Welch. Today's video is about protein synthesis, so making a protein. And for our cells, there are two main steps. Those are transcription and translation. So we're going to talk about the overall process and then each of those parts in more detail. So let's get started. Here's a stylized drawing of a cell that shows the overview of protein synthesis. So let's look at all the parts. This orange strand of little circles here is the protein. So once this detaches and goes off of this ribosome, that would be our protein, and that would be the product that we're looking for in protein synthesis. So the process is going to start here in this purple nucleus with the DNA, this blue double helix here. Transcription is just taking the message from DNA and turning it into an mRNA strand, transcribing, right, or kind of copying down that message into a smaller format. And then mRNA is going to go outside of the nucleus into the cytoplasm for the second step, for translation. And in translation, the mRNA, also shown here, joins up with a ribosome and creates the protein, or at this point an amino acid strand, and that will then be released, and little extra configuration will become the protein. So in my family, basketball is the sport of preference. I grew up watching lots of UK basketball. We even braved the blizzard of '94 to go to a relative's house with a generator to watch the UK basketball game when we had no power for over a week. So Dad's a pretty hardcore basketball fan. My husband's family is all about the Alabama football, so you can imagine our house. Now, you can talk about protein synthesis, this translation transcription, in terms of a basketball analogy or a sports analogy. Pick your favorite sport that has a coach and a team in the process. So looking at this picture, we've got the coach, right? And you know that in basketball, there's a specific spot for the coach to stand on the court. And if he gets out of that spot, then he gets a technical. So the coach, while he is making all the plays and he's directing the players, he's the boss, he still has a limited location where he has to remain. He can't just get out there and show them how it's done and play for them. He has to remain in his spot. And so this is like DNA. If you look at that DNA, it has to stay in the nucleus. It's too big to go outside of that organelle, and it's the big boss. We wouldn't want it traveling willy-nilly around the cell anyway. It has to stay in the nucleus. So the DNA is going to give the instructions to make a protein, right? It's going to send out the instructions for those plays, but it has to do it through a middleman because it can't go physically out to the location and do it itself. This is where transcription comes in. The first step where DNA is going to pass that message on to the mRNA to go out of the nucleus or out of the coach's box and go tell the players what's supposed to happen. All right, looking back at a picture of DNA, let's talk through that process of transcription, this first half. So this is just a picture, supposed to be a very simple picture of a DNA strand or a chromosome, a duplicated chromosome. And then you can see that as it unwinds, you get a little closer zoomed in here, you can see that double helix. Now think back to the basketball analogy. The coach does not need to tell the players on the court the entire playbook, right? He's not going to have a message go out and recite the entire playbook. He's going to say, a 5-step play, I want you to do this, this, and that's going to be the message he wants to send out at any one time. It's just a small piece. It's the specific instructions for what he wants to happen. That's the same thing that's going on with DNA. If you're going to make a specific protein, then you only want to send the message for that one little piece of DNA, not the whole entire chromosome. So we call that one piece of DNA a gene. So in this example, if you look at this little section right here, it's like we've unwound, right, this strand, and we're looking at just this little piece of it, and this would be the gene. This would be the one section of the playbook that tells how to make the protein that we want to make. The rest of it we don't care about. It's important to do other things. We'll use those plays later, but this is the only one that we're worried about, this one gene that encodes the instructions to make one protein. So there are three basic steps to transcription. The first is initiation. This double helix is unwound by an enzyme called helicase at the point at the beginning of the piece of that gene. And so this would be some kind of promoter. It would be a sequence of nucleotides that signals the enzymes that this is the start spot. So we have a promoter, that's the start signal. We unwind the strand with helicase, and then we get started. The second step is elongation, and this is where this polymerase comes in. Remember, all these aces means it's an enzyme. So RNA polymerase, they're represented by this kind of orangey bubbly thing here. It's a protein that helps get the nucleotides floating around in the nucleus, the right ones in to bind to the exposed nucleotides on this strand. So for example, the A here, remember that stands for adenine, and in RNA we don't have T's. So I guess backtrack a little bit. If you remember Chargaff's rule, Chargaff did some experiments and discovered that adenine always binds to thymine and cytosine always binds to guanine. So we say A equals T, C equals G. That's in DNA, but in RNA, we don't have any thymine. We only have uracil as its replacement. So the adenine would be on the original strand, and the RNA polymerase would help grab chemically a uracil nucleotide, a uracil-based nucleotide, so that those can bond together. And then it would move down farther, and it would have a thymine on the original strand, so it would look for an adenine nucleotide, and it would bind it together. So this would happen normally just without any help. Remember, enzymatic reactions always occur naturally, but enzymes help speed them up. So this is the polymerase is just helping the reaction occur much more quickly. It's helped bringing the correct molecules in. We say those are complementary nucleotides, the ones that bind to the other side. Now, if you'll notice, we are not binding to this bottom side. We are only binding to the top. So a gene is only on one strand. It's not on both. They are read opposite directions. So you may have a totally different protein or part of a protein on the other strand reading it the other direction. So you're only looking at one strand. So we're going to make a single strand rather than a double strand for this mRNA. Now, the last step is just termination. When we get to the end, the polymerase has matched all the nucleotides and then got to this end code that tells it this is the end of the gene. Then we terminate, and the mRNA is created and it's released. So we make a one strand of RNA, so it is a nucleic acid made of nucleotides. And it's released from the DNA, and then it can travel through the nuclear pores to go out into the cytoplasm to be used in translation. So that's all the steps of transcription. Pretty easy, just 3. Okay, so now the message is created and we're going to move on into translation. So think about it like that coach, that basketball example. The coach has decided what play is important. He's told one of the guys running by, right, hey, tells the, point guard, this is the play you're going to run. And the players are either going to go huddle up, or they're going to talk to each other real quick, right? And they're going to give those instructions to each person so that everybody knows their role in the play. And of course, those plays have a specific sequence. You've got to run to the block, you got to go left, you got to go right. And if they're done in the correct sequence, then hopefully you get that point. So translation, that second half, is where we take that mRNA strand and we're going to take that message from the DNA, take it out to the cytoplasm, and create the protein in the exact sequence that was told by the gene. Let's orient ourselves to this diagram and what all the little parts and pieces are according to where we've been so far. So this kind of palish reddish orange here is the mRNA. So this is what we made from the DNA in the first step of transcription. This is a ribosome, and ribosomes are lumped with organelles, but they're not technically organelles because they don't have a membrane around them, but they have a specific functions here. in this process, this is where protein synthesis will occur. So sometimes we talk about proteins are created on the rough endoplasmic reticulum, because remember, those are studded with ribosomes. So the ribosome is actually what's doing the process, but if they're attached to the rough ER, then that's a good site for protein synthesis. So ribosomes are made of two pieces that float around separately, and when the mRNA shows up, they join around it, okay? Another new molecule to mention is tRNA. So that's represented here by the green, and then by the blue here, they're showing it with different colors. tRNA is transfer RNA. Now, transfer RNA is helping bring amino acids. If you remember that a protein is built by, its building blocks are amino acids. So tRNA, transfer RNA, brings the amino acids to the mRNA to be joined together. So it's just a transport mechanism. Now these are very specific. Each tRNA has what we call an anticodon on the bottom. And that anticodon is a series of 3 nucleotide bases, right? So like this one's UAC. Whereas mRNA, we say these are codons. Every set of 3 nucleotides is a codon on the mRNA, so codon, anticodon. The other end of the tRNA has the amino acid, and these are very specific. UAC will always carry methionine. That's it. So we had to have very specific matchups here to make sure that we get the right sequence. Just like any set of plays, right? When they say all those code names that they've all learned, then they know what that means. And you can't switch the meanings midstream. You can't have one mean 2 different things, because then no one will understand. So everyone has to be very specific and carry exactly the correct message. So here's what happens in translation. mRNA shows up in the cytoplasm, or near the rough ER, and the ribosome, 2 subunits join together around it at the start codon. So if you see this start codon here, AUG, this is always the start codon. It's the only one that we have, and it always codes for methionine. So this is always the first step of every protein sequence. Now, once the ribosome binds around this codon, then tRNA can bring in and match up this codon, anticodon to the codon and bind. So look down here in this step, that's what's happened, right? They match, they are the complementary bases, and they are here. And then the ribosome can just move down another spot, okay? And it can move to the next codon and see what's coming up down the sequence. So again, it's like plays, right? You know, it's like first, you're going to go to the block. Second, he's going to go left. Third, you're going to go right. You see my basketball knowledge. And so the next one is UUU. That's the next codon on mRNA. So the next tRNA will be the opposite, AAA, and it will bind to N, bringing phenylalanine or whatever amino acid is next. Now, you would never have to memorize all these amino acids. There are 20, but there are 64 different codons. So we have some duplication there, and that helps us with mutations. So the next one comes in, and then this just continues. As we move farther down, so you see we've moved farther down, then that transfer goes, oh, well, you've got my amino acid now. I gave you the message. Go for it. And so as each tRNA comes in, it drops off its amino acid, and they bind to the next one, and then it goes off, and it can find another one of those specific amino acids to bind back with it, so it can bring another one around again sometimes. So we can reuse the tRNA. So you can see here we're bringing in the next one. Looks like it's GCU, and it's going to have an arginine on it. And as each time these are making bonds, we say those are peptide bonds as they are binding the amino acids together. And then this one's going to bind over here with this one. And you just keep stringing them on, like a series of instructions or pearls on a chain. And when we get to the end, there will be, just like in transcription, there was an end. We get to the end one, then it's going to say stop, stop codon. We have 3 of those. And the sequence is going to be complete. Everything falls apart, and we have an amino acid chain. And the cell can use Golgi apparatus and other things to package it and fold it up and get it in the right shape for use. Okay, that was transcription and translation. You might want to go back over that a couple times, practice those steps. Thanks for watching.